Thank you all so much for joining us this evening, especially getting here through the rain and the traffic. It looks like a parking lot out there, so thank you. I appreciate that. My name is Jane Wilkinson, and I'm the Executive Director here at the Koch Institute. And for those of you who don't know, what we do here is take a very unique and what I feel like a very MIT approach to solving some of the really, really difficult problems that we have in cancer. We are truly committed to convergent collaboration. We're always looking for new ways to detect, monitor, treat, and prevent cancer and other associated diseases. We've got an amazing community of biologists, biological, chemical, mechanical, material science engineers, chemists, and computer scientists. I really don't believe there are very many other cancer centers in the world who have taken this approach to doing this. You know, normally a cancer center is in the basement of a cancer hospital. We've actually built a dedicated cancer research center that really brings these different disciplines together. So tonight we're going to be showcasing our newest faculty and our clinical investigators. And I really believe that you're going to be meeting the future leaders of cancer research tonight. Now, unfortunately, due to a last-minute conflict, Kristen won't be able to join us, but we'll still continue with the program as we expected. One thing I really, really want to acknowledge is the philanthropic support that we have received to make the work happen here. And some of our supporters are here tonight in the audience, so thank you. I truly appreciate it, and our researchers do as well. Your support allows us to give the resources that they need to set up their labs here, to build their research teams, and to help them pursue their work and their dreams. And you're going to be hearing more about that this evening. But before we jump into the program, I want to take a very quick moment to talk about our clinical investigator program. So Joelle is here, and she's our new clinical investigator here at the Koch. And I really believe that our clinical investigators kind of keep us grounded in the clinical challenges, and they always keep us oriented towards the clinical solutions that we need to come up with. The clinical investigators program gives people like Joelle a really unique opportunity to take part in cancer research while still continuing their medical practice. And this gives us, the rest of the Koch community, a guiding light towards making sure that we're connected with the patient and with the clinic. And people like Joelle also give us a frontline view of the challenges that she faces when she's passing cancer treatment on to the patient. So we are going to hear from Francisco, Yadira, and Joelle. We're going to have 3 short talks highlighting their research. We're going to move to a panel discussion, and then we'll also invite questions from the audience. Now, for those of you on Zoom, I just want to ask you, if you have a question that pops up during the evening, throw it into the Zoom chat as soon as you think about it, because it takes a while for them to kind of come through and for us to collect them. So send your questions in on Zoom as soon as you have them. And with that, I am going to pass over to Francisco. Welcome.
Can everyone hear me well? Great. Okay, well, it's really an honor and a privilege to be here and have the opportunity to share some of the work that we're doing here, both in my lab, but also within the Koch Institute. And our work is mostly focused on trying to understand how cancers initiate, evolve, and what are the different events and genetic lesions that are required to do that, and how those types of lesions can influence the way that cancers are treated, with the goal of treating cancers in a more personalized way. And because as way of intro, I have to state that cancer is largely a genetic disease. That means that it arises from the sequential acquisition of alterations in genes that normally promote the proliferation of cells, or inactivating mutations that on genes that typically repress or inhibit the proliferation of cells. And each of these alterations in various types of combinations can lead to tumor initiation, which I am just diagrammatically showing here as an epithelial carcinoma. Now, because cancer is a genetic disease, I think we're actually living in a very exciting era for modern cancer research, because right now a cancer patient can go into the clinic, and typically, let's say the patient has a solid tumor, they'll take a biopsy of that tumor, they'll isolate genomic DNA from that tumor, the genome of these cells, of these cancer cells, and then they would sequence the genome, the DNA in these cells. And by doing so, they can actually identify specific genetic alterations that are present within that tumor. And the idea is that, and really an established model, is that those mutations that are observed in those cancers are the lesions that are responsible for initiating and also maintaining that disease, and potentially also affecting the way that these tumors are treated. So the idea that we can just go in now in the clinic, get tumor sequence, and use that information to try to tailor therapies is a very appealing idea. And this is commonly referred to as precision oncology or precision medicine. But the issue is that we can sequence very well. We can do this every day. We can do it multiple times, depending on the type of patient. And we can sequence more and more. But the problem is that until we know what each of those different genetic events, each of those lesions are actually doing to initiate and affect the progression and therapy responses of those cancers, we really can't make this precision medicine idea a reality, at least practically. And there's recent evidence showing that drugs that are designed to target specific mutant proteins like KRAS, for example, have shown promise in the clinic. And again, these types of paradigms really require us to know exactly what each of these different lesions, these different mutations are doing. And importantly, most of the mutations that are identified on a literally daily basis are variants of unknown significance. So that means that we actually don't know what those mutations are doing. Even if they are in a gene that we know the function of, say, KRAS, which is one of the most commonly mutated genes, we still don't know what each of the mutations are doing to initiate, maintain, and affect therapy responses of those cancers. And this is just a different way of illustrating what I just said. The details on most of my figures that I'm presenting here don't matter. The point here is that there are genes that are very frequently mutated in cancer, and they are mutated in many different ways. They're not mutated in the same way. They're not only deleted, they are changed in different ways that influence their function and also affect the way that cancers initiate and progress. And this is just another diagram where here I'm just showing you one protein. And the point of the diagram is that the y-axis here shows the total number of times within this specific data set that a given region of that protein was mutated. So the higher that bar, the more frequently it has been mutated. And the point is that these proteins, these genes can be mutated all over the region of the gene. So again, only understanding what one mutation does in this region will not necessarily tell you what a mutation will do in a different region. So we really need to take a holistic approach to try to understand what all these mutations are doing. And just as one example for this one gene, we need to understand whether all these mutations are redundant. That means whether they are actually doing the same. If not, what are the mechanisms through which these mutations act differently? And can we actually use this type of information to tailor therapies to specific genetic events? And there's evidence, as I mentioned, and again, just the details here don't matter, but different variants for a given gene, that is different versions of a mutant gene, can influence the way that tumor cells grow, respond to therapies. Patients that have specific mutations in a different gene here called AKT can respond to some drugs that are designed for that mutant protein, but not other versions of that mutant protein. And there are even more complicated versions of this where certain genes can be mutated in multiple places at the same time. And those events produce different effects than each of the single events, again, increasing the complexity of understanding this problem. So to understand and really interrogate these types of events, we've been developing and optimizing approaches that rely on genome editing technologies. You might have heard about CRISPR, and CRISPR is a very powerful technology that we can use to essentially target any region in the genome of cells or any living cell. But we're using a different version of CRISPR that allows us to go into that region of a gene and exchange just one nucleotide if we wanted to. So imagine you have a word and that word encodes for a gene that promotes or suppresses proliferation. We can go in and actually rewrite the sequence of that gene to model the effects of that mutation in causing cancer or in affecting the way that cells respond to therapies. So we're using these technologies, and this one is called base editing, but there are also newer technologies called prime editing, which allow you to do that even more precisely. And so just as one example, we can initiate different types of tumors in animal models that express specific cancer-associated mutations. We're essentially able to take a living animal, initiate a tumor that is driven by the same type of mutations that are seen in humans. So it's essentially the same allele identically, but now modeling a mouse. So we're actually creating a version of that type of tumor, and you can see it as creating a more personalized model for studying cancer. And this works very well for individual genes, but we also wanted to expand this type of approach to be able to interrogate the function of thousands and dozens of thousands of genes and variants that are detected on a daily basis in cancer patients. So to do that, we developed some approaches that allow us to do this in a large scale, high throughput manner. Essentially, we can take thousands of cells, each of which express a unique mutation that's seen in cancer patients. And then using assays that we can chat later, if you're interested in, we can assess in a population level the effects, the phenotypes that are produced by each of these mutations. And because of how well the technology works, we can then take, for instance, cells, a population of cells that expresses different types of alleles and treat them with different drugs to try to understand why, if some alleles affect the way that they respond to specific therapy, say drug A, but not drug B, and why some mutations might sensitize to drug B, but not drug A. And we can actually iterate this very efficiently in a high throughput manner. And so just as one example here, we did an experiment using this approach where we discovered new mutations in a gene that's really the most commonly mutated gene in cancer called PIP53. And the point of this figure is that introduction, engineering into the genome of each of these mutations allows cells to cause cancer. So this essentially validates alleles that haven't been discovered before this work and show that they're actually malignant alleles that promote disease. And we've done these experiments in vivo. This is just showing that if we engineer those mutations, we cause tumors in mice, and those tumors are driven by the same genetic events that are seen in patients. And then we've done something more complicated, which is what I just mentioned, where we do an experiment where we integrate thousands of different events, thousands of different mutations in a pool population, and then identify the mutations that are the most aggressive at initiating disease or increasing the proliferation of cells. And this is just one experiment looking at, again, thousands of different variants. And then one other, this is the last example, is that we could then identify specific mutations that have unique properties that loss of function or deletion of that gene does not have. For instance, this specific mutation produces a very metastatic phenotype that other mutations in the same gene don't produce. But typically in the clinic, a patient that has a tumor that has mutations in this gene called PIP53 will be classified as mutant, but one that doesn't have mutations in PIP53 will be wild type or normal. And what I'm saying here is that the mutant category has a lot more texture in a sense, because some alleles that are classified as mutant are actually much more aggressive than other types of mutants. And this is why we need to understand this. And this is just one example of how we can actually engineer this genome in a very precise manner. And again, happy to chat more about. And right now, the first two grad students in the lab are tackling problems in this space. One of them has, Sam has developed new methods to scale up this type of variant engineering to many thousands of alleles using different technologies. And Grace is interested in understanding how specific mutations influence the surveillance by the immune system of cancer cells, depending on the types of mutations that they have. So, and this is all coming back to the fact that every cancer patient is unique. And if we are actually going to use this approach of personalized medicine, we need to understand what each of these mutations do. So with that, I'll thank all the funding sources. The Koch Institute is a fantastic place, very supportive. Core facilities are amazing. And it's really a unique space to do research. So I'm very glad to be here and also very honored to give this presentation to you. Thank you.
Thank you, Francisco. We're going to hand over to Yadira and we'll take questions at the end.
Hello, everyone. Welcome. I'm sure that this is working properly. Welcome to the Koch, and it's really amazing to see the doors open and have, the community back into this amazing institute. So today I'll take you one layer above our genomic DNA, and Francisco did an amazing introduction to genes and how genes become broken in the context of human diseases such as cancer. But in our lab, we're actually interested in looking at what happens beyond genes. And that's the concept of epigenetics. So we're particularly interested in understanding how genetic and epigenetic based mechanisms of gene regulation are different, but also how they're working together to keeping really programs that are critical for our cells in check. And to start getting at this, we are focused on understanding and studying a structure that is known as chromatin. And for those of you that are not familiar with this polymer, this is actually the physiological form of all of our genomes. And in fact, our DNA doesn't exist as naked DNA. It's actually compacted by interacting with histone proteins, which are these very abundant proteins that are inside our nucleus. And then they form these particles that are known as nucleosomes. And they form these higher order structures that allow for our very long genomic DNA to fit in the very constricted nucleus space. And this structure is very dynamic in nature, and it has to be because it actually restricts but also allows the transcriptional machinery to come to the right genes to turn them on when they need to be activated. So we are really interested in studying how the epigenome actually is able to expand what the genome does. And the way that we think about this is that the genome is actually storing our genetic or hereditary information, but then chromatin actually provides an organization or an additional layer of complexity that really provides cells, and in particular cells in multicellular organisms, with an additional sort of plan that then you can activate these programs in a concerted manner. And in addition, this is a particularly important solution for multicellular organisms because all of us started as a single cell, the stem cell or fertilized embryo, that roughly has 20,000 genes that have identical sequences. So all the progeny from this stem cell will have the same genetic material. However, we have to go from this stem cell to over 200 different cell types that compose the adult human body. So how do you do this? And really the epigenome is providing what it's called as epigenomic diversification for them multicellular organisms to be able to arise. So you can imagine that these processes are very well regulated. And when issues arise, that will be causative for human diseases. And if you don't believe me, I will refer to a great artist, Lady Gaga, who actually, in one of her more famous songs, said same DNA but born this way, same DNA but born this way, really emphasizing the importance of epigenetics in creating diversity in human populations. So now we understand that the epigenome is actually regulated by a variety of multi-protein complexes. There's a whole plethora of these that have different activities that regulate chromatin states or epigenetic states. So for example, they're chromatin modifying complexes that basically what they do is that they catalyze chemical modifications into histone proteins, which I told you are the proteins that allow our genomic DNA to be compacted. They're actual remodelers. So these are energy consuming machineries, very large protein machineries that actually are able to move one nucleosome from one place to another, releasing sort of the compaction of our genomic DNA from, let's say, position A to B, so then genes can be turned on. And then there's also other structural factors such as chromatin readers that have high affinity for some of these chemical moieties in chromatin and also adapter proteins. And I'll come back to these factors in a minute. So we now appreciate how important chromatin is as really serving as a single transducer for both cell intrinsic but also intrinsic signals. So basically what chromatin does is it takes all the signals that a cell is experiencing and integrates them to then regulate critical cellular and molecular processes that include proper cycling through the cell cycle, so a cell can divide in an appropriate way, when a cell undergoes a DNA damage insult, chromatin is at the center of these DNA repair mechanisms. And also, I just alluded to this, it's very critical for proper gene expression regulation. And as I mentioned, when things go awry, particularly on chromatin, we have human diseases. And one good example of this is cancer. And we now think about cancer as really a cellular state of dysregulated gene expression programs. And Francisco really well illustrated that cancer is indeed a genetic disease, but it's also a disease that is characterized by chromatin and epigenetic dysregulation. And these two major processes can actually contribute to dysregulation of gene expression that then supports malignant transformation and then progression to tumors. So we now appreciate that chromatin and epigenetic dysregulation is actually a critical aspect of the cancer state. In fact, we have plenty of evidence now that suggests that epigenetic regulators are often co-opted or hijacked by cancer cells to support their malignant state. And something quite interesting that we started to realize over 10 years ago at this point is that chromatin and epigenetic regulators are actually among the most frequently altered genes in human cancers. So we're currently investigating what these mutations are actually doing in the context of cancer. And because of this, there's been a lot of excitement on trying to drug some of these arms of epigenetic regulation. And in fact, there's probably now at least one example of inhibitors that are able to impact each of these programs that regulate the epigenome. And there's still many outstanding questions regarding these epigenetic inhibitors. The first one is how do they work? What are the mechanisms of action? Also, what are mechanisms of acquired resistance to some of these drugs? And which types of cancers will benefit from these novel sort of therapeutic interventions? And lastly, are these going to work as monotherapies or will they be a better option in terms of combination therapies? So I mentioned that we are particularly interested on a particular subset of chromatin regulators known as chromatin adapters or scaffolding factors. And these are actually what the name implies. These are scaffolding proteins for formation of multi-protein complexes that have either modifying or remodeling activities. And they're quite interesting because they show what we call pleiotropic or content-specific nature. They can assemble complexes that can either activate gene expression or repress gene expression. And in particular, we have been focused on studying the prototypical chromatin adapter protein known as menin that we know is able to interact directly with histone-modifying enzymes, other structural proteins within chromatin, and also canonical transcription factors. And in the context of cancer, and really more relevant to this presentation, is quite interesting. Menin has been shown to be a so-called tumor suppressor gene or a stop gene in malignancies that arise from neuroendocrine tissues, particularly those that arise from the pituitary, parathyroid glands, the lung, and the pancreas. On the flip side, it has been shown to be an oncogenic cofactor or a GO gene in the context of cancer, particularly in leukemias that are characterized by genomic rearrangements that involve the MLO1 gene. And these are a particularly interesting subtype of leukemias because they are overrepresented in infant and pediatric patients, and they tend to be very aggressive. And to this day, there's really no targeted therapies to treat these patients. So really understanding the biology behind this particular subtype of leukemia will be very impactful for these patients. So just to give you a little bit of background of what we know about menin in the context of these leukemias that are characterized by MLO rearrangements. So we know that menin is absolutely required to bring to chromatin so-called MLO fusions that are basically a translocation product that occurs that includes the MLO1 gene, which I told you is a chromatin modifying enzyme. And because of this, there's been a lot of excitement on trying to develop, instead of going after the enzymatic activities of these complexes, now protein-protein interaction inhibitors. So basically, these are chemicals that bind to menin and basically abrogate the interaction with either MLO1 or the MLO fusion proteins. And you can imagine that disrupting these complexes could have a potential therapeutic impact. And in fact, these inhibitors are actually showing a great promise in clinical trials, and 2 of the leading molecules are currently undergoing phase 2 clinical trials. So when we started this project, we started by asking a very simple fundamental question. what are the cellular and molecular determinants that are present in these leukemia cells that allow them to respond to these anti-leukemic effects of these inhibitors. And through a combination of approaches, really taking very biochemistry focused, genetic, and also more mouse modeling approaches, we really made some interesting discoveries in terms of a mechanism that seems to have a very clear relevance in the context of patients. And I'll show you a few snapshots of some of that data. And I think this project really represents what we're trying to do here at the Koch Institute, to take very fundamental mechanistic insights into the clinic for the benefit of cancer patients. And we're hoping to continue working in this area. So in these two slides, upcoming slides, I'm just going to summarize a lot of work that will be the basis for some of the actual data that I'll show you in a minute. So the specifics of the sort of protein complexes really don't matter that much. What I want to mention is that this was the model that was established when we started the project. Menin is important for assembling a complex that is important to turn on genes that are critical for leukemia cells to progress in terms of proliferation and maintaining their malignant state. And that's what we're calling this sort of goal genes. What we found is that there's actually another portion of menin that is assembling a complex that instead of activating gene expression, is actually repressing gene expression. And in particular of so-called tumor suppressor genes. And these are the stop genes that basically when they're turned on, the cell is not able to proliferate. And then those are the mechanisms that actually keep our normal cells from becoming a cancer. So we found that this complex is also sensitive to the action of these menin amyloid inhibitors. So when you treat cells with these inhibitors, you're kicking off this complex and you're kicking off this complex. Basically, you can imagine that you're opening up space for other factors to bind to this part of the genome. And turns out that there's a second complex that actually turns on gene expression that comes in and binds where menin used to be and now turns on this tumor suppressor. And that was quite interesting because this work really allowed us to determine what is the mechanism of action of these menin amyloid inhibitors. But also, when we abrogated the function of this complex, we actually made the cells resistant to this drug. So it's sort of a dual finding in this case. And this is an important aspect for the data that I'll show you in a minute. So we wanted to take these fundamental sort of mechanisms that we uncover in mouse models of leukemia to more preclinical systems. To do this, we collaborated with a group over at the Dana-Farber that they have a very nice annotated bank of patient-derived xenograft models, particularly of AML, acute myeloid leukemia, which is the subtype that we were modeling in mice. And basically, we have two flavors of these PDX models. One that has that second complex I told you is important for activating these stop genes intact, and another one that has a mutation in the complex. So we transplanted mice with these PDX models and then treated them with a menin amyloid inhibitor. And this is here just to remind you the state of the second complex. And what we found is that when we treated mice with this inhibitor, we almost eradicated these leukemia cells from these recipient mice, which was very striking. And beyond this, when we take these leukemia cells, we actually look at their gene expression programs, and they're able to activate the genes that menin is regulating in this particular setting. However, when we look at a PDX model that has a faulty second complex, is not able to turn on these genes, these cells are completely resistant to the action of this inhibitor. And when we take these cells and profile their gene expression programs, they're also unable to turn on these genes, suggesting that loss of this second complex is really comparing resistance to these small molecule inhibitors. And lastly, in a very fortunate set of events, we were able to also collaborate with the clinicians who are running the trials for these menin amyloid inhibitors. And we got samples from the patients that are enrolled in these clinical studies to really understand whether some of these genetic or gene expression programs are being turned on in the way that we predicted from our mouse models. So basically, this is data from a patient who's responding quite well therapeutically to this small molecule inhibitor. So we profile these cells by RNA sequencing, which is a way where you can look for gene expression programs in an unbiased manner. And what we found is that patients that are responding therapeutically to these emerging drugs, they're also turning on the genes that menin is regulating in this setting. So you can imagine that this subset of genes can perhaps serve as a biomarker for response for some of these small molecule inhibitors. And with that, I would like to close by thanking my lab, growing lab, and everyone who contributed to the work that I presented to you today, funding sources, and in particular the KI, for being such a welcoming and supportive environment, not just to trainees, but to junior faculty who are coming and starting new labs and coming with crazy ideas, and you for your attention and support of this institute. Thank you very much.
Thank you, Yadira. And Joelle is going to give us the bridge from the bench to the clinic.
Thanks so much, and thank you guys for being here. I have to say it's really quite an honor to be considered up here with these junior faculty as one of the clinical investigators here. I'm a pediatric oncologist by training, and I opened my lab here earlier in the spring, and I had also spent some time training upstairs with one of our preeminent chemical engineers, Professor Paula Hammond. So I will say, first of all, that I'm so impressed and pleasant and pleased to hear from our biology colleagues about what to do with these pathology reports we get that say variant of unknown significance, because I will say that happens quite frequently. And so as much as we all hope precision medicine is going to be the case for a family member or a friend that we might know who has cancer, it's unfortunately not the reality yet for our patients. And so specifically thinking about children with cancer who I treat, we really need to do a lot more work to understand what's causing those tumors and how to treat them. But what I do at the Koch Institute is actually quite complementary, I think. My lab thinks about drug delivery. And so, just to give you a very kind of quick introduction to drug delivery, I'm kind of asking the how as opposed to the what. And so if you really think about what drives a cancer and what's leading it to be malignant or to metastasize to another part of the body, that's one of the things the biologists are expert in. But when you get down to what drug would you ideally want to get into every cancer cell and how would you get it there, you really want to talk more to an engineer. And so I'm trying to bridge that gap between clinician, engineer, and biologist, really by being at the Koch Institute. So I came here specifically to think about what drugs do we want to get to what cells in the body and how do we get them there at the simplest point. And so in this day and age, you may actually, we think about small molecules, which I'm showing here as little circles, as being the prototype of a drug, something like an aspirin or a Tylenol, right? But in fact, this day and age, you might be more interested in getting an antibody, something that specifically binds to a very specific protein, or even a cell, which you might think about CAR T cells or chimeric antigen receptor T cells that are then using your own body's cells to get somewhere and kill the cancer. And so all of these things are cargoes. And then the most kind of interesting cargo that you might have all experienced recently is a nucleic acid. And so I hope all of you in this room have benefited from the mRNA vaccine therapy that has really helped us to stem the tide of COVID-19. And so what you've actually received in your vaccine is a nucleic acid that is then complexed with some lipids into a nanoparticle that looks something like this. And so there's really a lot of ways that engineers and drug delivery experts can help you think about what drug that the biologists might have picked out or that the clinicians might want to get to their patients, and how can you put those into a polymer matrix filled with cells, or put your small molecule, attach it to an antibody to get it to the right part of the body. And there's so many experts here at MIT and all of the drug companies in this general area that think about these things that we can all collaborate with. So I'm going to focus today on nanocarriers and nanoparticles in particular, because it's an area of interest in mine and something that we're trying to bridge that gap between the clinic and the lab. And so to give you a very kind of brief historical overview of nanoparticles, why should you care about nanoparticles in cancer? The structure of a liposome, which is our prototypical nanoparticle, which is really just a little copy of the cell membrane that you can fill with anything you want, was discovered back in the 60s. And while that may seem like a long time ago, biologists have been at this many, many decades longer. So the discovery of things like bacteria and viruses actually predates most of this. So we still consider ourselves a fairly young field. But there's a few key dates I'm going to note here. One is 1995, which is when the first cancer drug inside a nanoparticle was approved in the clinic. And that drug is still in use in the clinic. And I think there's actually ways we might even think about repurposing it now. But then we had a big gap where there was a lot of interesting things happening in the lab and not a lot of things making it into the clinic. And with the exception of a few very exciting new approvals in the last 5 years, I think we're still not there yet in terms of getting nanoparticle drugs into the clinic. And it's not for lack of enthusiasm by our biologists and our nanoparticle engineers. It's really that translational gap of how do we get it over the line and into the clinic and find the right patient who's going to benefit from that. And so, this is where we're at today. If you look at just kind of over since 1994, when we had that first nanoparticle approved for cancer, and then all the way up to 2020, the new things, these little blue groups, are your 2 nanoparticle vaccines for COVID-19. And I will say that has sparked enormous interest from the general public, from the pharma companies, from all of the clinicians of, well, why can't we make that against my cancer or against my other disease? And in fact, I think there's a lot of opportunity to do that. But especially as a pediatrician, it's a little disheartening to me that only one of these approved drugs actually has an indication for pediatric cancer. There's many reasons for that, but it's actually an AML drug that's fairly effective called Vyxeos. And my goal for my lab is really, how can we make these numbers bigger? And it's not that I want to invent my own nanoparticle that I save for myself. I really want to think about how can we study nanoparticles in a way to apply them to a variety of tumors and think about what they're actually going to do for our patients. And so with that, I'm kind of starting my lab focusing on 2 fairly simple questions, which turn out are very difficult to answer, but this is a great place to do it. And so the first one is, how do nanoparticles cross physiologic barriers? And the most key barrier that we think about for most nanoparticles is the blood vessels. So you might get a drug injected into your vein. It has to circulate all around your body. And then in particular, getting to brain tumors, you have to get across a specialized blood vessel. The second question I think about is really almost more of a zoom out broad question. How do cancer cells see nanoparticles? If you treat a variety of cancer cells with a variety of different nanoparticles, how do they all interact? And I think those 2 things together will hopefully give us the design tools we need to design better drugs for the clinic. So I'll give you 2 short vignettes today and start first thinking about how do nanoparticles cross physiologic barriers. And so to think about this, it's really, what do we see in the clinic that helps us to know what's important for our patients? And so my actual particular expertise is in pediatric brain tumors, but there's a lot of relevance to adult brain tumors and other tumors that metastasize to the brain as well. And so one thing that is abundantly clear to any patient who might be in the cancer clinic with one of these, and also to the oncologist, is that getting drugs to the brain is really hard. Patients with brain tumors do not do as well as patients that have that same exact tumor growing in a different part of the body. Once a tumor has been able to metastasize to the brain, it's unfortunately very difficult to treat. And so whether you have a tumor that arose here in the brainstem, like I'm showing, or a tumor that maybe arose in the lung or the prostate or the breast and then went to the brain, or have a leukemia cell that has made it into the spinal fluid and is now coating the area around the brain, these are very, very difficult clinical challenges, and in fact are some of the most difficult conversations I have in the clinic. And what it really comes down to is that we don't understand why the same drug that works in the liver doesn't work in the brain. And we know that the blood-brain barrier, or the specialized blood vessels between the brain and the rest of the body, are the problem, but we really don't understand how to get past them yet. And so, we like to think that everything can be broken into boxes, and I also like that. And so, I'm just showing here quickly what might happen. If you take a drug, it goes into your vein, so something like an intravenous injection, you first have to have that drug all around your body and your blood at a certain concentration. Then you get to your central nervous system barriers, which is more than just the blood-brain barrier, but all of these specialized blood vessels that allow things to get into your brain, your spinal cord, your cerebrospinal fluid, the fluid coating your brain. And then even if that maybe 1% or less gets into the brain, it might be cleared very quickly. It might have trouble getting into the tumor cells that you actually want it to, and instead go to the healthy brain cells. So we just have an abundance of barriers. But really, at the end of the day, we need to get enough of the drug to the site of the tumor to have an effect. And so starting at kind of the simplest state of that, we need models in the lab that help us to develop these drugs. And so a project that I worked on with Roger Kahn's lab over across the street, also here at MIT in mechanical engineering, along with Cynthia, who was the lead grad student, was building a model of a blood-brain barrier on a chip with a cancer cell. And so we really wanted to have something in the lab that we could look at, that we could flow things through the blood vessels and get them out to a brain tumor. And so we did that by taking cells that are important for the blood-brain barrier, and actually a patient-derived tumor that came from the very generous donation of a patient and family. And we put these things together first in little tubes and then into this tiny little microfluidic device that would be about the size of a quarter. And then after about a week, we're able to look at this and say, you know, how are the tumor cells in the blood vessels interacting? How is the tumor growing in the presence or absence of different treatments? And we can also ask some interesting biology questions that I would then need my biology counterparts to help me answer. And so just to give you a quick look at what this looks like, I don't know how well the red projects here, but we're seeing here in the red, these are all blood vessels that you can fill up with different fluids, and then the green is the tumor. And you can actually see individual tumor cells are kind of coating and touching the blood vessels. And this is something we imagine is happening in our patients, and I'm sure it is, but at the level of a surgical procedure, you don't get this level of detail. This is something, you know, this 100 micron here, that's maybe a thick hair from your head or your beard. You know, this is a very, very small piece that we don't see when we're in the operating room. We don't see with an MRI scan of the brain. And so our hope is that by building these models, we can then use them to test the drugs we want to deliver. And in particular here, we were interested in looking at nanoparticles. And so to give you a very brief primer on nanoparticles, we make it sound very, very easy, but I will say that there are many students here working really hard to make these things happen. And so I don't want it to diminish it in any way. But what we did here was make a liposome. This is what I would consider like a very simple nanoparticle analogous to what's in the clinic. And then we coat it and we put things on the surface. Here we're using natural polypeptides, so kind of like very, very tiny pieces of protein that we can control and engineer that are non-toxic to the body, but probably contribute to how the drugs might be released and move around. We can wrap these things using what we call layer by layer technology and then use a targeting moiety on the outside. And so this is another peptide or a very short piece of protein that is able to interact with receptors that we know are expressed on the brain vasculature. And so actually this chip allows us to answer a couple things. You know, what proteins are expressed on the blood vessels close to the tumor versus far away? And are our nanoparticles that we designed to get there actually doing their job? And this was a really nice example where we could actually show that the nanoparticles can cross the vessels close to the tumor, but they don't cross the vessels far away from the tumor where the protein expression is different. And so our hope is that this type of model for brain cancer might help us to develop drug carriers that can get into the cells you want them to, but avoid those healthy developing brain cells, especially in a pediatric patient. And so we're really hopeful that others will adopt this model, that we can expand it to many other cancer types, and then also look at many different drug carriers. And so going to the second question, and I'll be brief, I hope I'm doing okay at time. How do cancer cells see nanoparticles? This isn't a question we might have been able to answer, you know, even 10 or 15 years ago, because the technology to do really high multiply parallel pooled sequencing wasn't quite at the point where it is now. But we really wanted to answer this question: what is it about a cancer cell that makes it take up a nanoparticle or not? And so what we did here was we used pooled cancer cell lines. And so I'm going to show you just some kind of cartoon graphics. But here I'm showing all the different nanoparticles we designed and tested. And most of them were using that same layer by layer technology that I talked about earlier that allows us to coat the nanoparticles and control very carefully the chemistry that's on the outside. And then we had about 500 different cancer cell lines, and each one contained a little barcode. And so it's really similar to think about a barcode at the store. It's not changing what the fruit or the vegetable is, but it allows you to check it out with this little machine. And so we had these inert barcodes inside the cancer cells. And by putting these two things together, we could then say with a really high throughput, you know, cell line A works really well with nanoparticle B, but cell line C doesn't. And to ask some questions thinking about things in a really, really big scale. And so, you know, what questions could we ask with this? One, and the most simple of them, is what matters more, the middle of the nanoparticle or the surface? And in fact, when I started studying nanomedicine, we all thought we had this figured out. It's the surface. The surface has to be the thing that matters. That's what the cells see first. And it has to be the most important. And that was the assumption. And in fact, by using this layer by layer technology, we could really ask these questions and say, we're going to show the nanoparticle, show the cell this exact same surface with two different cores, or we're going to show the same core with all these different outer flavors. And the answer was actually quite surprising to us. And so when we looked at how similar all the nanoparticles were to each other, that's what this big red line down the middle means. They were each perfectly similar to each other. We actually saw that the core mattered more. So in this case, lipid-based cores or fat-based cores with these liposomes all acted more similarly to the same cancer cells than two nanoparticles that had the exact same surface, but a different core material. And so perhaps we weren't giving cancer cells as much credit as they might have due to them, because cells apparently are able to see the core and the surface at the same time. And so this has changed a lot of how we think about what nanoparticles we should be designing for what patients. But even more than that, we can ask some pretty complicated questions. And so I'm not going to go into details here, but a really great part about doing these large pooled cell line screens is that we know a lot about cells. And in fact, people like Yadira and Francisco have generated a lot of data about a lot of cells. And so by taking omics data, which is the entirety of the genome or the entirety of the proteome or the entirety of the epigenetic modifiers of a cell, if other people have generated that data, we can plug it in. So with plug and play techniques like that, we can then say, all the cells that performed well with this nanoparticle all had the same gene expression pattern in common. And we're starting to now unlock a little bit of, what can we look at about a cancer cell that might predict which nanoparticles are going to be most likely to work for that patient. So we're at the very, very early days, and this is kind of brand new data here, but our hope is really that by understanding how nanoparticles cross physiologic barriers and get into cancer cells, we can then kind of team up just at the right time with those biologists, discovering which drugs they want to get, and then work with the clinicians to get them to the right patient. So with that, I will say it's complicated. This translational gap of how do you get something from the lab into the clinic is really quite difficult. And many of my clinical colleagues have found it, quite disheartening. But I actually feel the opposite. I feel like when we get together with biologists, engineers, and clinicians, there's a huge opportunity to tackle this if we can bring together systems biology or kind of these more complex biology features, and then also keep the patients in mind of which patient might be most likely to benefit. And so thanks for letting me share a little bit about my lab. We have a growing small lab, but I also work very closely with Paula Hammond's lab upstairs. And I wanted to thank all of my funding sources, and thank you for your time.
So I'm going to ask the speakers to come and sit up front with me. We are way over time, but we did start late. So if you can bear with us for about 10 minutes while we do some Q&A, is that okay? Right, thank you. You can be the rose between the thorns. Thank you all so much. I'm going to pose our first question in darkness. No. Our first question, I'm going to kick off to Joelle. I thought we'd go in reverse order here. So as a practicing oncologist and with somebody who's running a research program here at the KI, how do you bring that clinical experience, that patient interaction to the bench? I mean, you've talked about it with your research and how you're thinking about how we get those small molecules into the patient. But on a day-to-day basis, how do you kind of guide your research with your clinical interaction?
Well, I actually have to say, I think the biggest impact I might be able to make is probably not through my own research, but either by training the next generation or actually working with my colleagues. So I can't tell you how many colleagues have asked me questions that might be simple or easy for me to know, how many patients have this, or is it likely to see a mutation in this? Have you ever had a problem like this in the clinic? Things that I also love to talk about, but then they can take it back to their labs. And so I find that there's always an intersection of everyone here with the clinic, and there's always a way to kind of make those connections even stronger. So whether it's connecting a colleague of mine who studies a rare tumor with someone who studies a similar unusual mutation, or whether it's actually using patient samples that are from our tissue bank and starting to analyze them with the more complicated spatial omics techniques that we have here, it's just too easy to make the connections in many ways.
Thank you. And to Yadira and Francisco, how do you maintain the importance of us always having to think about going from bench to bedside? How do you balance your scientific curiosity with the clinical importance and the clinical outcome. Because, as scientists, we all know it's super easy to go down our favorite rabbit hole, but still to stay focused on that clinical outcome.
I can start. So I think my training as cancer biologist by Bradley training here at the KI actually keeps me grounded and reminds me why we're doing what we're doing, because I got trained into more molecular biology, biochemistry, which, as you said, takes you into those rabbit holes of what is the answer, what is the answer. But I think I'm still able to pull myself back and think about kind of the greater question. What is the impact of what I'm thinking about, the questions I'm asking in a very fundamental way, how they can help patients, right? And I hope I illustrated some of that with the work I presented today. It started as a very fundamental, basic question. And it had, we tried to put all the pieces together, but still keeping in mind that the question was to the benefit of these patients, perhaps to improve, the development of combination therapies or predict resistance mechanisms. So I think it's almost inherent in my mind, perhaps like training. But yeah, it's very important.
Hi. So for me, very similar to what Yadira mentioned, but I actually studied microbiology as an undergrad and was studying microbial ecology, but I always wanted to do research that impacted human health, and that's how I eventually transitioned to cancer genetics. And I think one way of that keeps me myself grounded when I'm thinking about a lot of the technologies that are out there and the things that we can do, which by the way, should really blow everyone's mind that we can actually re-engineer genomes at will using these technologies. I have to remind students that we can actually do this, and it wasn't possible 10 years ago. So even when I get deep into the weeds of that technology, I'm always thinking back as to now we can actually use these technologies to finally understand how we in our bodies have the genes that become mutated that lead to cancer development, and how these genes influence therapy responses. And I think it's important to keep that in mind, but also to be in the right environment. And I think having people like Joelle, where she's saying, we have these drugs, not everyone responds, and then someone like me asks, okay, why are they not responding? Is there anything genetic that we can test in the lab? And then we go and test those. But I think that this is the perfect environment to do that, because I did my postdoc at Sloan Kettering, which is of course a clinical, very clinically and treatment oriented. And I found myself thinking more and more about translating what I was doing in the lab. But I think you can also push it too far and not think about what you can go back and do in the lab somewhere like here in a multidisciplinary environment where you can actually have longer term impact instead of just thinking about the like how can I treat a patient tomorrow. And that's this balance of keeping myself grounded and thinking about the how the work will impact is what keeps me going, and the environment is a big part of that.
And just looking to the audience, please raise your hand if you want to ask a question, but I do have one question that's just come in on Zoom. I'm looking at Joelle, but I think we might all have an opinion on this. How does nanoparticle size affect drug delivery efficiency, especially when we're looking at different tissues, different organs, and drug retention at those particular sites as well?
Yeah, it's a very good question, a very technical question. And I like it because I think, to be fair, we haven't been as rigorous in things that have gotten to the clinic. And this goes for nanoparticles as well as small molecule drugs. When we do a clinical trial and we find that something worked or something didn't work, those clinical trials haven't always been designed to show us why they didn't work. And so there's actually been quite a few nanoparticle trials that pass phase 1, which is about safety, but they don't do well in phase 2 or 3, which is about efficacy in more heterogeneous or kind of usual populations. And I actually think this question is right on. We don't always know, which organ for which patient is this nanoparticle really getting to. But on the flip side, if a nanoparticle is properly designed, it can release its drug at a set and known amount of time and you can control having multiple drugs inside the same nanoparticle. So there's only one example of that in the clinic so far of a combination, 2 drugs inside 1 nanoparticle, but I think those are some of the ways we probably need to move into the future. But we do need to be very rigorous about understanding where the drug is releasing and how it's going.
Yes, question. I'm going to run over to you with the mic.
Okay, perfect. So my name is Zainab, and I'm a biomedical engineering at BU student. And I have a question for you, Joelle. What would you say could it could be the reason the core protein was core lipid was more important than the polypeptide that was surrounding it?
Yeah, it's a great question. So thinking about, this core and surface. And so there are ways to potentially oversimplify it as I did, but it is true that having, 2 different surfaces will certainly affect the extent of nanoparticle uptake. So for instance, in a given cancer cell, 2 proteins or peptides presented on the surface will change that extent. But when you zoom out and look globally, everything with a lipid core is acting in a more similar way than everything with a polymer core. And to me, it speaks to the lack of inertness, I will say, of lipids. And so to explain that, lipids that have encapsulated drugs are actually usually considered by the FDA to be an inert part of the drug. So you're getting the drug approved wrapped in a lipid. But in fact, lipids aren't inert. They're part of our cells. And in fact, we do see that nanoparticles with different lipid compositions are interacting differently with cells. So I think we may use that to our advantage, and you could think about weaponizing your nanoparticles with the right lipid that's going to bring them in. But the polymers, and they're maybe all a little bit more bland. So I think the lipid specifically might be a biology question.
I'm just looking at the clock. I'm going to close out. Oh, there's a one hand at the back. Oh, that's a long way to run. You would think that with it being MIT, we might have better technology than me.
Hi, I'm Parag Mehta from a company, Avita Biomics. I had a question for you that I totally understand in glioma for a drug to cross the blood-brain barrier is a major challenge. But as a practicing oncologist in pediatric gliomas, to what extent do you think that challenges about drug delivery and or actually just lack of effective drugs, the molecules themselves, that can actually address the molecular heterogeneity of the tumor? Because even you are able to get the drug, 1 or 2 drug molecule inside the brain, but the tumor heterogeneity is so much, and then tumor constantly mutates. So these are the fundamental issues for getting a durable response. You might be able to get a response that may extend a lifetime for a few months, but then what do we do about actually the fundamental issue of finding the right kind of molecules? And more diversity of molecules you get, more challenging is going to be to deliver them.
Yeah, you've hit on a big, I would say controversy, but it's really more of a discussion amongst brain tumor docs, I would say, across the country and across the world. There's kind of 2 camps. One is we haven't found the right drug. The other is we've never gotten the drugs into the tumor at the right concentration to do their job. I fall, of course, a little biased on the one side, but we certainly need combination therapies. We've never cured any brain tumor patients with one drug. And so we need combination therapies, and we ideally need to find those that can do different jobs inside the cell and actually hit different mechanisms. So I think we need better biology to understand, which drugs go together, but then we still have a delivery problem. But you're right, it's a difficult problem to solve.
Should have put my sneakers on.
I just have a short question. I'm also a clinician, radio-oncologist, and of course, we've been speaking about drugs a lot tonight, but there is something interesting about radio-oncology, and that is that, especially with regard to biologicals, we have sensitization. We see that a lot, just the reactions on skin and so on. If somebody's taking a biological and we irradiate, we have rashes, reactions that are undesirable. But it might be an interesting thing to study what concentration is actually necessary, especially considering the abilities that we have with stereotactic irradiation of microscopic tumors and things. What concentration is actually necessary? How could it even be possible to determine the concentration in vivo? These are questions. I have a little a memory that tumors that exceed about 5 millimeters the blood-brain barrier is damaged and the drug can get into the tumor better. I don't know if how well that's been studied.
Yeah, no, we should follow up after. So I'll certainly find you to discuss some of the nuanced things, but certainly we don't really know, and each patient's tumor is probably different in terms of how the blood vessels are changed. But it's absolutely true that most of these small molecule exciting drugs we give, even if a little bit of it gets to the tumor, a whole lot of it gets to the rest of the body. So maybe that's something we all need to think about. You know, how are these targeted drugs affecting our healthy skin, affecting our healthy lungs and liver? Because yeah, we do see toxicities with these new small molecule agents that are sometimes hard to manage.
Thank you. I'm going to close out with one final question for Yadira and Francisco. So here at the KI, we're extraordinarily entrepreneurial. You know, we have pushed out over 130 new companies because it's essential for us to translate our discoveries into a company so we can actually get to a patient. You know, MIT is never going to be a drug company. MIT is never going to be a medical device company. What new technologies are you most excited about, and what are you looking forward to in the future of your research?
I guess I can get started. So I think for me, and I alluded to this a little bit during the introduction, is the development of these epigenetic drugs, but in combination with other agents like immunotherapy, for example. And I think we're seeing some great success stories in the clinic right now. When you are controlling things down, you know, at the epigenome level, but at the same time, you know, interactions with the immune system, I think you're hitting a lot of angles that cancer cells cannot escape. And I think to me, that's really going to be the future, particularly for the pathways and the mechanisms that I'm interested in studying in the lab, and really how these epigenetic drugs will be working in concert with other treatment modalities like immunotherapy.
In my case, I probably will be biased, but again, I think that these technologies that we have now, in theory, should allow you, if they're efficient enough, to correct some of the mutations that are disease causing. And there are some examples already out there in I would say, significant clinical trials, advanced clinical trials that have shown that you can actually do permanent correction of certain genetic diseases, mostly liver-associated diseases because of delivery being easier. But I think that that's one area that I think it's definitely transforming already, and you can potentially fix these mutations. But also, as I presented, they also allow you to interrogate all these different genetic events across many different scales, across many different tissues, and you get a lot of data. So the other big advance that I think is also very strong here at MIT, and it was actually one of the main reasons why I wanted to come back here, is really what do you do with all this data? And once you generate a lot of data, you could generate predictive models that allow you to then predict which patients might respond to different drugs depending on the combination of genetic events that they have. So people like me and my lab are producing this type of data that I think will be perfect to be integrated with computational biologists and machine learning experts to try to create models that allow you to predict and better rationalize and stratify treatment options. So I think the integration of those two will be one of the advances that I'm most excited about.
We're going to close out. I just want to mention a couple of things before we close. First of all, thank you so much for being a part of this and sharing your passion and sharing your research with us. We have a couple of upcoming events. We have a special talk from Professor Richard Hines, who just recently won the Lasker Award for his work on integrins, and that's going to be happening on November 2nd. We also have a book launch about Salvador Luria, whose auditorium we're sitting in. That's coming up soon as well. But one thing I do want to say before we head out is that if there's anybody who wants to connect either with the researchers here tonight or with me or wants to navigate the Koch, please reach out, please drop me a line. You can find us all on the internet. We're very transparent. You can find our emails. So please reach out to us and also join us outside in the gallery for some drinks and nibbles. Thank you.